Okay, so now we have to take that theme forward, I guess, in many respects. Well, what are the drivers of a lot of this innovation and change going on? So, whilst the panel take their seats, I'll just set the scene a little bit, if I may, and introduce Priya Kalia as the moderator. I've known Priya for some time, and I think what really stands out to me is the fact, A, she likes to communicate, and B, she likes to challenge around that innovation edge. And I think what we've heard there from Melanie really is about we have to be thinking more broadly, but actually then thinking with focus as well. What are we actually trying to say and achieve? So I'll hand over to Priya, MD of SciTribe, and going to take us through understanding what's driving us. Priya.
Thank you. Is the mic on? All hear me clearly? There we go. So yes, welcome to the first panel discussion on Helix. I think myself and everyone here, we're very excited to be here and to kind of share our thoughts on enabling bioinnovation. What are the barriers? What are the exciting things that have been happening? And I think the Keynote presentation really helped set the scene. It was really fascinating to see and complements the conversations that we've all been having over the past weeks. So yeah, I think the first thing I think that would be useful is for each panelist to introduce themselves, a bit about themselves, the companies that they are representing, and their thoughts on bioinnovation and impact. So, David, if we just start at the end and work our way up.
Hello everyone, hopefully the microphones are working, they are, great. I'm David Joyce, I work at Thermo Scientific. I work as Director of Product Management for the Connect digital platform. Been working in the industry for 25 years, mainly around LIMS, scientific data management, and integration of instruments and extracting that data and making sure it goes into the right places. We've been building, well, I've been doing this for quite a while. It was the paperless lab before, and then it became Lab 2.0, and then now we're talking about this bioinnovation. I look at it from a fairly simplistic point of view, in that there's probably three things we need to at least get right before we can start to do the AI and ML that we're all looking to do. Our samples, they need to be well identified, they need to be traceable, you need to be able to track them from one place to the next and know their locations, and do that in a consistent manner. If we all look at the way that we're doing it now, I've seen so many freezers with notes written on them, and oh, so-and-so knows where that sample is in boxes in frozen up freezers. Instruments, we make it so difficult. I mean, as an instrument company, make it so difficult for you to manage those things. Preventive maintenance, you've got to get on the phone and ring up for that. That should be done. Part of the platform that I work upon is connecting using IoT, so that these instruments are sort of ringing back to the home base and sort of making sure that they're always working, always available when you need them. But then also the translation of the samples from your systems into those instruments. We make you write that down or type it in or transcribe data into the instruments. That should be seamless. And then finally, the data that comes out of those instruments. That it needs to be exported in a way that it can be used by the downstream stream systems. We like to keep our data proprietary. I don't know how many formats, even across my organization, we have for that data. Once it comes from the system, how do you tie it together again? So the themes of sort of interoperability and sort of data standards, I think that was the thing that we all shook hands on when we were talking from hardware to software. Having data standards that allow you to link this data together, rather than brilliant minds spending days and days in spreadsheets trying to align the data before they can even start to do anything like AI and ML. Now, if that needs a human to do, how can AI ML even work? If you need to line these things up in a manual fashion. So a bit of a soapbox for me, really, but I'm glad they brought me onto the panel. And over the mic, I think.
Okay. Thanks. Yeah, and just to mention here, we've tried to, in the panel, encapsulate all the different parts of kind of maybe the life sciences workflow through from the data generation with the right, with advanced technologies and instruments through to the data management side with with David, and then with BC Platforms, the data science angle. So with that in mind, Michael.
Hey, I'm Michael Chen, co-founder and CEO at Nuclera. We're about 100 strong at the moment, based both here in Cambridge, UK, as well as in Boston, Massachusetts. So what we do is we make proteins accessible by launching the world's first protein desktop bioprinter. And what that desktop bioprinter does is really unites this disjointed, fragmented, and labor-intensive protein accessibility workflow by combining express, characterize, and purify of proteins on a single desktop bioprinter, single device in a single day. And we're able to do that by combining our tech stacks of cell-free protein synthesis reagents, protein detection chemistries, and other assays directly on our digital microfluidic cartridge, which we developed in a strategic partnership with e-ink, which is the pioneer of e-paper displays that are found in every single Amazon Kindle. Turns out there's a lot of similarity between the technologies required to make displays and the technologies required to make digital microfluidic devices that we use on our desktop bioprinter. And so we believe that, together with others, this entire panel, we're part of this wave of companies that's really trying to accelerate the digitization of the wet lab and to really both save time and bandwidth to our drug discovery practitioners. So if you have any protein problems, you use proteins within your discovery workflow, check us out at nuclera.com or feel free to come up to me during the conference and talk with me. Jeroen?
So my name is Jeroen, co-founder and CEO at Semarion. We don't focus on proteins, but on cells. We've created a novel materials platform whereby we can bring adherent cells into suspension. So why are we doing that? Adherent cells are basically the workhorse in drug discovery to screen compounds against a variety of cell models. And while there's been a lot of innovation with suspension-based cells through innovation with, let's say, liquid handling, ultra-miniaturization, think about 10x Genomics or Berkeley Lights, for example, similar innovations haven't happened really for adherent cells. What's more is they suffer from long preparation times, not a lot of flexibility in the assay workflows, and also reliability issues. So that's why me and my co-founder came together and really leveraged technologies from the microfabrication industry, the semiconductor industry, and apply novel innovations in the material space to solve these biological problems. And we've created a technology which we call the SemiSite assaying microcarrier platform. And these are essentially ultra-miniaturized wells that can carry sort of colonies of adherent cells into suspension. And we can sort of integrate this with existing workflows and equipment to accelerate sort of cell assaying workflows and methodologies, making it more reliable, faster, and really, like we were saying earlier already, take away a lot of pain points for, let's say, cell biologists and drug discovery people to study quicker, more resourcefully, and faster. And again, I think this is going to be a theme that's going to come back over and over again during the discussion. But we all sort of agreed yesterday that we want to free up the brains of the great scientists out there to focus on drug discovery or diagnostics and other sort of areas of innovation, rather than, in my space, for instance, endlessly pipetting cells and caring for cells. So I think people could use their time more wisely. So that's why we're here on the panel today, I think.
Great. It's a pleasure to be here in Babraham. My name is Paul Smith. I'm part of a company called BC Platforms. I'm the business development director of BC Platforms. I remember 25 years ago, just about 100 yards there, we spun out one of the earliest Babraham companies, Life Science Resources, very successfully. So it's lovely to be here. So BC Platforms, we're a Swiss-based company, but we're investing heavily into the UK. Why are we investing in the UK? I suppose with the ex-Boris Johnson, if nobody's heard, Boris is resigning today. but one thing he did do very well was kickstart the life science strategy, which I'm sure all of us are very much aware of, which was kickstarted about 4 or 5 years ago. And really was all around maximizing our brilliant 60 million cohort population, which we have in the UK. This is the biggest cohort of data that is in the world. It's recognized as the biggest cohort because it's located in it's essentially an infrastructure of the NHS. This globally is recognized as very, very powerful. So we should recognize that. So BC Platforms, we're helping to facilitate the access to that data and securely and in a sort of federated role. So we're leaving the data where it is, where it's being produced, either from the instruments here. But with unique technology, we're able to access that data through this federated approach. I think you were talking about the trusted research environments. Trusted research environments are going to be becoming incredibly important in the UK. We're pushing with the bleeding edge of that again in terms of making NHS data available to researchers outside of the NHS and even globally. We have fantastic cohorts of data, UK Biobank, the 500,000 project, which was kickstarted 15 years ago. Great genetic information, great omics data, and what BC Platforms is bringing all that data, omics data, linkages with genomics data together, but not together as one central database, but federated. So we're bringing the analytics to the data. So technology has made that accessible, made that easier, and you're not having to move data, large amounts of data like genomic data around. You're moving the analytics to the data in a secure and federated approach. But glad to be here and glad to be in Cambridge. Yeah.
Thank you. Thanks, everyone. So hopefully that sets the scene for the discussions, as everyone's referred to, that we've been having around bioinnovation and current, I think from everyone's perspective here, current trends. I think one thing that came up time and time again, there were some key themes. So people, and I think there's been agreement that really AI is not replacing humans anytime soon, and they are an essential part of the whole life sciences innovation. With regards to data and standards, this being a very important point for discussion, in terms of enabling bioinnovation, and also the role of automation. So I think I'd like to, I think the thoughts about people really link into also data analysis, data access, and data standards. So I think then if we look back at that kind of subtopic, what do you think? If we, start with you, David, would you like to share some thoughts on that?
We started talking about people, and then we went off on a complete tangent around sort of how hard it is to hire people in the, after the Great Resignation and what have you. A lot of the work that I've been doing with companies has been sort of to make the environment for scientists so much better. You have to attract people now. If you're going to go take them somewhere where they have to sit there with their slide rule and their piece of paper to do the work, then they're not going to be happy to come stay with you. A number of the big pharma companies are building new buildings and integrating their instruments with their data capture capabilities to make it's removing the digital drudgery. I coined that phrase yesterday, but essentially to make it a much easier environment to work in from a data point of view. And again, that talks to standards and standards of output, and that took us down the path of how do we get to standards? I mean, I've been working on the animal subcommittee in I think it's ASTM. It's been going on for about 20 years now, really, to try and come up with this all-encompassing standard for data in the analytical space. Still not there.
And why aren't we there, David?
Well, it could be something to do with the people, the type of people that are on that type of committee. I was working with another standards body, which is OPC, which is OLE for process control. It is prevalent across all of the manufacturing space. And it was the benign dictatorship I talked about. Essentially, they came up with something that was good enough as a standard, and it got rolled out because it was practically used. It was put into industry, and it was 80% there, and then they've iterated on it. There's a different approach. I think from a scientific point of view, we like to be 99% pure before we put anything out into into publication, whereas the engineering approach was more around sort of let's get to a practical minimum, get that out there, and actually start to use it. You mentioned a standard that is sort of being used, and it is the standard, but it's because it was practical, it was useful. What was that?
So on our platform, the first standard that we report that's pretty standard in protein sciences is soluble expression, yield, identity, and how soluble is that protein that you're expressing. And you can go down the solubility multiple ways. You can either do a relatively inexpensive but labor-intensive experiment, or you can do an incredibly expensive and time-consuming experiment. So it's to me, the theme across the panel seems to always come back to accessibility, right? Whether that be AIML accessibility, protein accessibility, cell accessibility, population sequencing accessibility. So it's how do we launch the technologies, devices, tools, reagents, et cetera, in order to make the life of that drug discovery practitioner, the scientist, that much easier. And that's why the theme at Nuclera is always designed by scientists for scientists. How can we make the life of that benchtop scientist as easy as possible, so that we really do free up bandwidth of that scientist?
Yeah, absolutely. So, I think the reduced drudgery to data point has been one theme. I think it's interesting what you mentioned, Michael, about the kind of data that can be produced for solubility, let's say, and going back to the engineering kind of take on the whole matter, which is what is good enough in terms of data standards, what is good enough in terms of data access. I don't know if you could comment a bit further on that.
Well, the reason why my co-founders and I founded Nuclera is because we all had issues with the inaccessibility of biology generally. So I'm a structural biologist by training. You don't hear a lot anymore. You hear more about AlphaFold, AIML, drug discovery, et cetera. But as a structural biologist, the bottleneck I had was just getting the protein necessary to put into my crystals trays. And that literally took me 4 years to get. So it was really the accessibility to proteins and the ability to work with them in a quick fashion and iterate on that protein accessibility workflow. That was a problem at the benchtop for us. So to me, data access is being able to get the tools necessary to get the physical materials so that you can actually get that data point quickly and in a cost-effective manner.
Okay, fantastic. Thank you. Jeroen, did you have some thoughts on
Yeah,
this?
I think we're talking a lot about sort of like the data side of it and how we can annotate the data in a way that different people can use the data at a high standard of quality and do some machine learning on them or mining the data for whatever you want to do. But there's also an issue around the interoperability of equipment. And that's what we focus on quite a lot as an organization as well. We've developed our materials so that they can integrate with existing workflows and equipment, because when we talk to, let's say, our pharma clients or biotech clients, what they don't like is people coming in with a new bulky set of equipment, because they already have so much pieces of equipment sitting around, and real estate is expensive and they don't want to reinvent their whole workflow over and over again. So there's lots of players coming in with new solutions that require bulky new equipment, which is one issue. But then the second issue is that all these pieces of equipment don't work together. And if we're talking about the lab of the future and, let's say, cloud-based labs and solutions, these machines really need to work together. And to be able to facilitate that, again, that might come down to standards, samples, et cetera, but also the software piece in terms of how can you control those various pieces of equipment. And I think there are some people potentially working on that nowadays as well. And that's something that I'm quite excited about myself. Yeah.
One of the things that I thought was almost criminal, you were saying, oh, well, we've got this fantastic technology, but we're going to concentrate on this tiny little thing because that's, we've got to do one thing because we've only got a certain amount of resources. So we'll build the software for that particular specific, the low-hanging fruit or something like that. And in the meantime, all of that potential is sitting there on the shelf because you're the only one who knows how to program the software for your particular instrument. And we talked then about sort of this idea of having open standards and opening the APIs, application programming interfaces. If you publish those, you see great things happen. We've got to be a little bit more open about our technology, at least as far as data access, such that a third party could come along and start working on your platform before it's even out of the shop. So that's what the standards tend to bring.
And I see that trend happening with all the major life science companies. We talked about Thermo Fisher, GeneArt, Danaher's IDT has APIs that plug into everywhere. So does Twist Biosciences, and these are right at the beginning of life sciences DNA synthesis, right? You need to be able to send your sequences to these companies to even begin your experiment. And to me, interoperability is the ability to unite workflows with technology, either on our eProtein desktop bioprinter or with technologies like yourselves.
And we've certainly learned, haven't we, that you can't operate in isolation. If you're doing proteomics, genomics, imaging, all of this data needs to be integrated into one. They all have valuable facets that they need to add to the solution, if you like. So you can't do anything in isolation or in silos. They have to be interoperable. You have to bring all the data and look at all the facets of the data, and all the pharmaceutical industry companies are doing this in terms of their drug discovery, from biomarker identification all the way to clinical trials. So incredibly important, and also incredibly important that it's linked with clinical patient data, because you can't do any of this. You can't really analyze any of this data unless you've got the kind of clinical connection or linkage to all of this data.
So I wondered, Paul, if you could comment a bit more on data access and interoperability as well.
Yeah, I think data access is incredibly important. We all operate in a little bit siloed in terms of researchers. We tend to create research projects, have certain projects that you keep the data. But that data is incredibly valuable, whether it's the quantity or quality of that data. Some of this data can be very small, but actually is incredibly important. So rare disease or cancers, and this linkage between all these different data sets is becoming incredibly important, and is possible now because we've got the technology, we've got cloud that essentially helps this with IT infrastructure. So it's a fantastic world because the technology is around to be able to do all these exciting new things.
And in light of the pandemic, there has been a lot more sharing and access, and that was mentioned earlier. So if that's already happening, what should that not be continuing? And
well, the pandemic was fantastic, wasn't it?
Yeah.
Especially in the UK, because
Write that down.
certainly for the UK. But in a space of 9 months, we managed to get a vaccine out, and that was all around 24/7 access to the data. So we had the researchers, the clinical trials, we had the regulator, all looking at the platform 24/7. Which facilitated this, increase in efficiency, reduction in the drug discovery process. The regulators are now looking at this because it's obviously saving huge amounts of money. We're getting drugs quicker out, and that's all around this access to the data, making it available 24/7.
Do you foresee any risks to that model? As again, Melanie kind of mentioned earlier, do you, from your perspective, do you see any potential closing off over time?
It's interesting. We've got the skill. We're sure we've got the skill, but have we got the will now? Are we going to go back in and be retrenched back into our sort of proprietary, this is my data, sort of situation? How can we foster that spirit of Dunkirk spirit, COVID spirit across industry? I'm not sure I'm seeing that.
Right. Interesting. So.
Yeah, and I think the biggest thing is this sort of patient. What we get engaged with was with the patients, and what we need to really be very careful of is security of their data, isn't it? And everybody's slightly paranoid about it. But if you do talk to them individually, they're willing to help research, and they're willing to engage, and they're willing to put data onto their app or make that data available. And if you can do that, if you can talk them through that kind of consent model, the world's going to be a better place. And you can see why there's paranoia and things about data and security, but really there's mechanisms in place now to make data secure, anonymized, pseudo-anonymized. And I think patient engagement is incredibly important in this process.
It'd be interesting to take a bit of a straw poll of people in this room, because they represent the people who would be asking to put their data out there. How many people in the audience put their banking data online?
Oh, yeah.
In fact, how many don't? Okay.
Yeah.
Oh. Fair enough. But so you do that with your banking data, and a lot of us put our stuff out on Facebook as well, our personal data. But then, how many would put their scientific data out onto a cloud platform? I have to say, it's a lot fewer, and I think there's a real sort of suspicion in there, because data is the ultimate product. Discovery, the ultimate product of what we do. We're very insular about that. I mean, we were talking about thumb drives and hard drives and my data on my PC. I mean, as an IT guy, it's proven that isn't as secure as actually putting it into Amazon's cloud, where they pay very clever people to look after the data. But I'm the same. It doesn't feel like it's yours. It doesn't feel like it's secure. And I think there's a psychological barrier we've got to get over. I find that in every case. I've got people who don't want to put their pH meters online because they're worried about the data getting out. It's like, really? Why is it? But you put your presentation, your PowerPoint, onto Microsoft Office 365, which is out there on the cloud with all the conclusions and the data.
Yeah, absolutely.
Without any compunction.
Well, I think that's changing really quickly, right? Because just 20 years ago, you wouldn't think about willy-nilly sending your DNA sequences out there, which effectively tell everyone what targets you're working on. And that's exactly what people are doing. Mm-hmm. Twist, GeneArt, ID, it's all in the cloud at the moment. So it's a step-by-step. As you see, we deal with a lot of large pharma companies, and we understand IT is one of the most conservative areas, but
it's a matter of building trust, I think. So, I mean, you were pointing out about sort of GeneArt and the amount of information that, if Thermo Fisher were evil, we could be mining that for our own benefit. Obviously, we don't do that. Just write that down. But truly, I mean, that there's unintended consequences. I think we need to even have the legal terminology. I think you probably at the forefront of this, that data is secure, and there are safeguards legally as well as technologically that you can trust your data in the hands of others.
It's what our marketing head always says: platform credibility. Right? We're launching a lot of new technologies out there, and it just takes time to get an ultra. I mean, this industry is ultra conservative largely because of the infrastructure set up around it, including patents.
Yes.
Right. The entire patent, we can't share what we're going to talk about for fear of getting scooped. We need to file our patents first before. So there is a bit of that in the siloed culture of the biotech industry, but it really takes technology, innovation to maybe open up a little bit, and some reform in the way we file and protect our technologies.
Yeah. So who's going to lead that?
That's a good question.
Please raise your hands.
Yeah, I mean, at any point during this discussion, we welcome input from the panel. Davide.
So I wondered whether it's a cultural problem or an incentive problem. Like if you compare Melanie's point earlier on about the silos academics, if you compare the point before on the presentation about the silos academic with the sort of productization of the device, which is an obstacle per se, with the sort of data privacy issues. I mean, there is a sense that we're not always operating for a common good, and this creates a fear, sometimes paranoia, but I think it has to be recognized as legitimate fear.
Yeah.
But I'm wondering how much of this three different problems is cultural, and how much is actually the incentives not being aligned?
Thank you. That was really, really interesing. So, should we take another comment and then maybe let you respond to that?
I was just thinking about what happened.
If you could just speak up a little bit more.
Oh.
Yeah, thank you.
Hi, Paul Rampage from Transcript. I'm just wondering if we could take the leaf out of what happened in the gaming industry in terms of using common platforms, and it released a huge burden of new games because people didn't have to reinvent the wheel.
So actually, that's my job now within Thermo Fisher in some respects. We have, Thermo Fisher, we go out and we pick and choose companies and bring them in and integrate them. But we integrate their platforms. They get a nice Thermo sticker on them, and but every one of those companies comes in with their own software, and it's what the Americans like to call a mom and pop shop. So all of this data security, the formats, even the graphical display is all done from scratch by teams such as your teams, if you like, and it's always done really well. People are quite attached to it, and then you try to integrate that with something else within our organisation. It's really difficult. So we've got initiatives within the organisation to sort of do the basics centrally. So central identity platform. So you log in with your own login from your own business to thermofisher.com in our case, and we federate the identity from your system. So that's something that they can use as a building block. We've got data scientists who are working on components that we can use for data science. And it kind of builds almost like an operating system for people to then put the smarts on top of. That's what our Connect platform is all about. But it has to be open because Big Brother Thermo can't be the only one that's doing this. We'll end up with a Danaher platform, and we'll end up with an Agilent platform. I'm sort of not from the instrument side of the world in Thermo, and we know that it's got to be an open platform. So we'll publish the APIs. We use data standards, and I think when we participate in data standards bodies like the OPC UA Foundation and Allotrope, so that the next generation that we build isn't going to be in those silos that we're talking about. But that's a really good analogy, actually.
So I have a question then. Do you think it's going to be one of these large companies is going to be the winner in the end, or is it going to be still a fragmented market with lots of different standardized, let's say, APIs and platforms that companies like ourselves and scientists can plug and play from?
I think it's going to be the latter, and it's an uneasy truce. That we want to get you onto our instrument platform. We want to tie you in with our proprietary formats. That's ending, and it'll come from the requests from the people in this audience saying, "No, it's my data. It might have been made on your instrument, but it's my data. It needs to be in a format that I can transfer to somebody else's system in order to do it." And I think we need to be a little bit more savvy about what we ask for, because what you ask for, you'll get.
That's really helpful. So I think it'd be interesting to hear your thoughts, Paul, but also response to Davide's points as well around the validity of some of the concerns and alignment of incentives, and also what is cultural and what might be otherwise.
Yeah, I think you were talking about who's going to drive it. I do think, again, with the COVID aspect to it, I think the regulators are looking to drive this and make sure, in the drug discovery process, that pharmaceuticals are a bit leaner and more efficient in terms of their drug discovery process, which is incredibly conservative over the last 30, 40 years. So maybe it's going to be driven right from the top, from the regulators. And, we all came together during COVID.
Yeah.
Maybe that's the positive aspect to this, that they will drive these making things efficient, making data efficient.
How do you mean the regulators have driven that? I've got battle scars from 21C.
Yeah.
Where they said, we'd like you to do digital electronic records, but we're not going to prescribe how it's done. Go to it. And we wasted tens of millions, hundreds of millions of dollars.
There's an example in the pharmaceutical industry at the moment in terms of using external control arms, for example, so data that's outside of the pharma companies and using that data. So it makes it more efficient. They can stratify that cohort more efficiently with genetic data. So it's really dollars and cents. They really think, the drugs are getting more and more expensive. That's the driver, is drug development much more efficient, much more cost effective, and that hopefully will drive the whole pharmaceutical industry in terms of this whole data interoperability and more efficiency.
What I really like is, so I was pointed at Boehringer Ingelheim's Open.me website, where they share the target. They don't share everything, but they share a lot of the targets they work with, some of the hits or leads that they have in their pipeline. That to me is an example of where the industry could move and be a little more open. And ultimately, there's definitely an incentive to be paranoid and keep your stuff internal. But we're trying to solve really big problems right now. For example, we all saw Moderna go from sequence to authorization in 11 months. How do we replicate that across the entire drug discovery industry? So the reason why people are thinking about adopting our platform, which is a protein synthesis device that links to the cloud, is because of the promise that it can help them get, or be one of the solutions that gets to target to hit in months rather than years, right? So it's uniting around solving some of these grand challenges, I think, that will drive the industry towards being more open, more collaborative.
Absolutely. I think the grand challenges is a good focus. Good point.
What I also wonder is whether or not we need a central arbiter of the data to some extent, because we're talking about patient data, we're talking about discovery data, and then sharing it. But once you get access to that data, you trust it is of a high quality. But that might not always be the case. I mean, lots of papers are not even reproducible. So how can we trust that the data which is shared, which is again going to be the foundation of further research, if that's scientifically sound? And from patient data, maybe that's less of an issue. But if people start to culture their own cells or produce their own proteins and then do a whole study on the back of that and then share that data, well, how do you trust that that data is of a good quality? And that's, we were talking yesterday a little bit about quality systems for data and sort of annotating whether or not data is of a good quality or not. But that's a whole other sort of like subject we haven't really touched on yet.
I would love to hear some thoughts on that, and then some comments and questions from the audience would be nice. But I think data quality was
Maybe to go back to Moderna, if you hear Stefan Bincel talk, he says one day he wandered into a laboratory and saw one of his scientists working with sequences in Microsoft Excel, which is absolutely horrible. And I know scientists at Nuclera still do that because that's comfortable to work in Microsoft Excel, Word, to handle sequences. And you have no what are you doing, relying on spell check to make sure these sequences are accurate? So there are tools out there, but I don't think there are any unified tools to help. And that's a critical aspect of data quality is maybe removing the human a little bit from the input and handling of that data.
Okay.
Yeah.
Great.
But please don't let the last word be removing the human from the
Because as we've discussed, humans are incredibly important. So
please, someone else say something.
So actually, this does touch on something we've been discussing, which is the role of the human in future, having increased bandwidth from these amazing tools and increased use of application of automation. So what is the role of the human in bioinnovation?
So where we got to in our conversation, it's interesting the analogy with the gaming industry, but in software now, we have agile development. Everything's done in 2-week sprints, and as a product manager, that's brilliant. I can see, this is what we've built. Oh, that's terrible. We need to go in this direction, and we can change direction very rapidly with that type of technology. We're not really there. When it takes you how long to culture your cells and then whatever. Yeah.
We tried to adopt 2-week sprint agile methodology, and all the scientists internally said, how the hell do we do anything in 2 weeks?
But if we can take the drudgery out of it, if you've got to spend all that time culturing your cells and I love my Gilson, but doing the pipetting and whatever,
No more pipette sales, yes.
E1 clip tip, it's not Gilson.
That's right. Always got to plug Thermo.
But there is an awful lot of that grunt work that is done by brilliant minds. If you had a platform where you could actually This is what I'd like to do, go do it. And you can automate a good deal of that, and actually iterate as well. We've seen a lot of automation platforms, and we had the amplitude system during COVID where we string and sealing wax together something that would do 8,000 tests a day. But that's a one-trick pony. It would only do that. I think where these chaps were talking about is some of the technologies that they have allow us to have one of those high throughput systems, but then you can tweak it incrementally, tweak the hardware incrementally, and change the methodology sort of dynamically, rather than it being sort of, automation is always a 2-year project. It could get down to the point where it's a 2-week project to change that automation. I'd love to see sort of a confluence of some of the things that you're bringing together with the automation, and then some software that makes it easy to do that.
So yeah, it'd be great to hear just a little bit about that. Maybe just one thought each, and also where's the role of the human in that, in that iteration and in that process, sorry.
When we talk to our partners in the US, in Europe, a lot of the peers that are developing technology solutions for iterating at the benchtop, together with others, I think we're really at the cusp of, in the next 5 to 10 years, having thought to result workflows and devices directly on our benchtop, being able to have that group meeting, talk about what experiments you want to do, and then next day having the results directly to iterate rapidly, which doesn't exist today in the marketplace.
In the life sciences, perhaps we're in biotech,
Exactly.
but in tech, I think
Exactly,
we were
and
discussing that you do have these models in tech, and how behind is some of what we do compared to the tech
Exactly,
industry.
and so a lot of VCs in Silicon Valley will say, we're the biotech is the next tech, right? And we're in the 60s or 70s of tech. But I like that analogy a lot, because that's basically saying, well, ultimately, the life scientist will become like a computer scientist sitting behind a computer and doing code to perform experiments. And the theme is, well, how do we digitize the wet lab experiment? Because all scientists here know that in silico is not good enough yet. You need to do and be grounded by that wet lab experiment.
Absolutely.
Yeah, I enjoy tremendously from working with my co-founder, who is essentially an engineer. And I think, David, you touched on this earlier as well. The mindset is so drastically different. I always try to design my experiments in a way that they would take months and months and months and be of a super high quality. But his mindset was more of iterate fast and really get to a solution that you can implement as fast as possible. So this is sort of like a mindset change that we might see in bioinnovation as well. And I think we all hope that with the technologies that we're developing, that we can facilitate that by freeing up people's minds away from the bench into sort of like thinking about methodologies that can be implemented and executed rapidly in a more, let's say, iterative manner rather than with long sort of cycles in these. And there's probably room for both. Of course, once you're doing drug development, it needs to be scrutinized to a high standard still, but those early phases can potentially be sped up a little bit with a less sort of wasteful sort of strategy. Yeah.
Perfect, thank you. At this point, yes, there's someone in the audience at the back, and it'd be great to hear your question or comment.
Oh, yeah. Is the microphone working? Hi, Kat from DomainX here. Just thinking about how we can move away from sort of the siloed culture, as you call it, and enable us to learn from what's going on in other industries. Do you think that there's a place for sort of cross-industry events? We tend to have meetings where we share the room with like-minded people. Maybe we should mix it up a bit so that we make sure that we are exposed to things that are out of our comfort zone and we are inspired by what's going on elsewhere.
Yes, I think that's an excellent point. I, for one, very much believe in that kind of idea. I don't know if anybody checked out the CogX festival that happened last month, but that was very much a confluence across industries, kind of driven by tech, but it was definitely bringing people together across different industries, something to check out, I think. But it would be great to hear a few thoughts from the panel.
Well, I sort of drew my analogies from the process industry. I mean, they solved a lot of the problems that we're trying to solve probably in the 60s and 70s. And they realized that certain stuff is pre-competitive. Instrument data formats. Why are we making a separate mass spec format to Agilent and to Waters? There's no need for that. And it doesn't really give us any advantage. So I actually think that again, it needs to be powered by the consumer of the technology, so yourselves, to sort of say, look, that's not good. Let's have a standard. Let's bring that data together. Even then, we can speak the same language. We've still haven't got to deal with the security and the regulation aspect, which is where I'm interested to talk to you afterwards about what's being done with those data sets and how you're securing them and making them accessible.
Yeah, and I think you were mentioning healthcare, IT companies looking at healthcare as the next future. And I absolutely, Google, Microsoft, Amazon are all looking at this. DNA sequencing is one of the largest data sets in the world. I think Google was saying it's similar to YouTube now. So they see the kind of commercial aspect of facilitating healthcare and pharmaceuticals. So engagement with IT is incredibly important now, and so Cambridge is lucky to have Microsoft here and R&D. So the more you integrate or talk to these guys, the more they're interested in. And the other thing that's machine learning, artificial intelligence is something that we've all talked about a little bit, haven't we, forever? But I think we're getting really to the crux of making those models, testing those models on large data sets is becoming accessible and proven now. So a lot of that will filter into our industry. But yeah, engagement with IT, I think, is incredibly important.
And it sounds that if we don't work with Dell and Intel, they might do it without us.
Yeah.
Indeed.
Yeah.
Excellent point. So I think on that note, if there was one, we have one person at the back. Maybe if we could squeeze in two.
That's me. Oh, sorry. I thought there was somebody behind me. My name is Madhu. I'm from LifeArc. I'm just asking, now we have started investing and having fostered innovation in the rare disease space. How could the community come together to enable innovation such that rare disease patients can have better outcomes, better therapeutics reaching the clinic? And I'm asking this specifically because as we are beginning to look at data sets and how they can help inform through, say, synthetic controls, are there any thoughts from the panel as to how to make this happen?
Right, thank you. So I think, also just off the back of the things we've been discussing today, I mean, what are
Your lanyard, maybe.
Is it my lanyard?
Yeah,
Oh, sorry. Do we have a response, like a quick response as well? Because I know
I think there are
we're supposed to
people
move on.
in this room who are working on repurposing drugs, right, to treat rare diseases. So not having to go through that rigmarole of preclinical all the way to phase 3. Additionally, it's really part of the trend of making drug discovery quicker, cheaper, faster in all respects. And it's a really long and complicated process. So everyone needs to work from that preclinical discovery stage, target to hit stage, all the way through the companies that are innovating in clinical trials with software and in silico tools. So it's not easy, obviously, but it takes a lot of people working at it.
Okay.
So, were you talking more about how can we better collaborate with your question? Yeah.
2 points actually.
Yeah.
Making it more visible. The challenge is more visible. Clearly the commercial challenges are very clear at the small market. So how could we use and combine what's happening in the market where there are already therapeutics to actually use those technologies, data solutions, etc. to actually also in parallel enable innovation to reach
I think it's actually an interesting point. There's a company that lots of people will be familiar with, Helix, and what they've tried to do is use data to sort of like shortcut a lot of the sort of steps that you would normally take for the drug discovery cycle, because of course there's less money available, so you need to do it more resourcefully. So you need organizations like that who want to take a central sort of position to do exactly that, and then work with data providers, technology companies, and integrators to essentially achieve their goals in a more fruitful and rapid manner, I think.
That's great, very helpful. There was one more point at the back.
My name's Rick Patel. I'm from MedCity. Really interesting discussion, and I agree. I think looking to learnings from other industries is going to be really key, especially in life sciences, where I'm sure everyone can appreciate it can be pretty siloed on that front. So I think shared learnings is going to be equally important. But looking forward, and I think the discussion has been quite top down from this moment. I was a bit curious from your perspectives on the sort of bottom-up aspect and kind of looking at the academic partnerships or instilling that confidence and trust from the up-and-coming generation and maybe creating some sort of stewardships, because looking at a loose parallel, thinking in disruptive innovation as well, going back 10 years, Facebook, that was just an online platform for students. Look how far it's come in 10 years. And it's really changing every industry, as we know at the moment. So I'd like to get your perspectives on how that sort of collaboration with academia could help to influence and implement alongside that top-down approach to enabling bioinnovation.
Yeah.
Thank you very much, Tracy. Just to say, if we could get some thoughts, and then we don't want to keep everyone from their coffee break. So I think we'll make this the last.
Yeah, last question. So, actually, interestingly, I think that the people who are coming up through academia at the moment are much more open to open source and open systems. The vast majority of the 200,000 people who are connected to our Connect platform are academics, and they seem to sort of use it throughout their academic career, and then the paranoia sets in when they get into the industry, and we're seeing a real drop off. We don't see them once they've moved into industry. So if we can actually capitalize on that attitude and give industry the confidence that the data is secure on this type of platform, I think we will see that sort of grassroots approach to working together on these platforms.
What I'll say as well is it's important to work with academia, but when your board and your investors are saying target pharma, target biotech, academics always want discounts. It's difficult. So and it's expensive to launch these programs with academics. So it's important to have platforms, programs to encourage that. And so I point to a specific program we're involved with in Belgium with VIB. They have an alpha and beta program and grant funding dedicated to helping companies launch their devices into researcher labs at VIB, and they fully fund that themselves. And so that provides the funding for small companies like ours to be able to launch those devices, not for free. Copying that, taking inspiration from those types of models is going to be very helpful, I think, for the UK.
Yeah, and I think from the other side as well, industry and big pharma, for example, they obviously love academics as well, because it's the academics who generate novel technology, potentially spin out a company, generate IP that they want to acquire, license, and these kind of things as well. And it's really about creating ecosystems that facilitates that communication, because of course the earlier that communication starts, the better the solutions are that are being generated. And that's with industry, but it's also with clinicians and patients, for example, as well. And what we need is more, let's say, institutes or organizations that facilitate that communication, where people have a platform to communicate openly with each other and not hold back, potentially come up with some small pilot studies that they can do together. I think a lot of people are very scared of doing collaborations with industry, because they think there's going to be these long term sheets and NDAs, and that really is sort of a block or a barrier to that innovation piece. And if you can remove that barrier by facilitating small little experiments, I think that can open up a lot of possibilities.
Yes, and I think, thank you, Jeroen, that's a great point. I think it also relates back to the question we had about rare disease and trying to get to patients sooner as well. So thank you. Any last thoughts?
Look, I think academia is incredibly important, and you can see companies like AstraZeneca moving into the centre of Cambridge just on that basis. It's that connection with the university, the cancer research, the hospital. They want to engage with academia as much as we all do, and academics, researchers are driving this kind of industry. So, Astra have really come into the heart, physically into the heart of Cambridge, and it's all around, as everybody knows, it's usually in the pub, isn't it, that you have a chat with somebody, or the Eagle, or wherever, where we can transfer information between academics and pharmaceuticals, and we should do more of that for sure. But the Astra having a physical presence here really gives Cambridge that. The university is incredibly important to us.
Thank you. So ending off on a very interesting point there about physical presence and interacting with each other as we move on to our coffee break. So thanks everyone. Thank you. Thanks to the panel. They've really worked hard on trying to make this a good conversation. And just pass back to Toby.
Thanks, Priya, and to the panel and to Melanie earlier. I think if one thing I took away from this morning, there was part of it that reminded me 20 years ago when my kids were 4 or 5 years old and they used to watch this thing called Bob the Builder on telly. And there was this big question, can we fix it? Well, their challenges of fixing some animated road hole or something, or pothole, weren't as complex as some of the big challenges that Melanie set out and we've talked about here. But I think COVID has probably shown us through collaboration and maybe being broader minded in who we engage with. Or maybe for some of those challenges, and then the answer is yes, we can. Or you can, I can. But with that, time for a fix of caffeine or sugar or just to rehydrate, and we reconvene at 11:50. Thank you.